17. A sick chicken leads to the discovery of cancer-accelerating genes. In 1909, a Long Island poultry farmer brought a Plymouth Rock hen that had developed a conspicuous tumor in its right breast to Peyton Rouse, a young medical researcher studying cancer in animals at the Rockefeller Institute in New York City. The farmer, referred by his local farm bureau, feared that this might indicate an infection, such as chicken cholera, that could threaten his entire flock. Dr. Rouse took a biopsy and determined it was a malignancy of the muscle known as a sarcoma. Seizing the opportunity to work with a cancer that had spontaneously arisen in an animal, he initiated research directed toward finding the mechanism through which the tumors in one animal might be transmitted to another, in the hope of preventing it from happening. His first step was to implant a small piece of the tumor into another hen of the same species. To his excitement, a sarcoma developed. In a series of classic experiments, he ground the tumor tissues to an ultrafine consistency and passed the material through silica filters with pores so small that they would eliminate all possible cancer cells and bacteria. As the cell-free tissues still caused the disease, he assumed the cause to be a virus. The concept of a virus at this time was poorly understood. A virus was seen as an invisible poison that seemed to have a life of its own. One Rouse was able to transmit the tumor through several generations of hens. He was the first to demonstrate that a virus could cause a malignant tumor. Rouse published his findings in 1911 in the Journal of Experimental Medicine, the official publication of the Rockefeller Institute. The scientific community scoffed at his theories, as the prevailing dogma held that most cancers were not contagious or the result of infection. Experience had long implicated environmental agents, such as chimney ash, but how this or other chemicals might cause malignant tumors remained a mystery. Some cancers were known to run in families, but there was no understanding of the hereditary factors. These revolutionary findings were generally disbelieved, Rouse recalled decades later in a 1966 interview. His peers believed that either the growths were not tumors or the agents were tumor cells the filters had let through. Discouraged by the lack of support, Rouse soon abandoned these experiments, bitterly deeming cancer research one of the last strongholds of metaphysics. The idea of a viral cause for cancer was, for the most part, put into the mental attic of researchers for decades thereafter. It was not until the 1940s that the Rouse sarcoma virus was identified by electron microscopy. As 20th century medicine progressed, researchers came to better understand viruses and even came to attribute viral origins to some tumors, as well as to leukemia. In 1955, the first issue of the new journal Virology appropriately carried an article on Rouse's work on chicken sarcoma. 55 years after his breakthrough discovery, at age 85, Peyton Rouse was awarded the Nobel Prize in 1966. The insight, the pregnant hint, as Walter B. Cannon liked to say, had to await a favorable intellectual climate and technologic advances. To reproduce, cells and many viruses must copy their DNA into RNA. The Rouse sarcoma virus, like some other tumor-causing viruses, was known to consist not of DNA, but of RNA. As early as 1963, Howard Temin conjectured that this virus's lifestyle was to insert itself into a host cell's DNA, where it would reproduce viral RNA. This idea, revising the accepted concept of the flow of genetic information from DNA to RNA, was largely rejected until 1970 with the independent discovery by Temin and David Baltimore of reverse transcriptase, an enzyme that directs such an operation. Such viruses came to be termed retroviruses, and it was speculated that the viral RNA in a host cell not only facilitates the virus's replication, but might remain dormant until expressed as a cancer-causing infectious agent, perhaps activated by external carcinogens such as radiation, various chemicals, or even other viruses, in effect, commandeering the cell's genetic machinery. It was amid this scientific ferment that Bishop and Varmus came on the scene. In 1970, Harold Varmus, who was interested in the genetic basis of cancer, began working as a postdoctoral fellow in the laboratory of virologist and biologist J. Michael Bishop at the University of California at San Francisco, UCSF. Following up on Rouse's newly appreciated work, Bishop and Varmus set out to study the cancer-causing gene within the Rouse virus. Harold's arrival changed my life and career, Bishop said. Their relationship soon became one of equals, and they would make their major discoveries as a team. Viruses are subcellular forms of life, basically mere packets of genes, generally fewer than a dozen. In 1975, Bishop and Varmus made a startling observation. The virus they were studying had somehow appropriated a gene that sparked malignant growth in the host cells of chickens. The Rouse sarcoma virus contains only 4 genes. 3 of these are used to reproduce the virus. The 4th is the gene that induces the cancerous growth. They had come upon an elemental secret of cancer, a gene that can switch a cell from normal to cancerous growth. What was the source of the gene? Does a cell itself harbor such genes? A piece of the mystery unfolded, like lifting a corner of the veil, in Einstein's famous phrase, when they found that the gene was present in healthy chicken cells as well as infected ones. The gene was not a native component of the virus, but rather at some point during the virus's cellular passages, either as it moved from cell to cell in one host animal or as it passed from one chicken to another, it had picked up an RNA copy of a chicken gene. The California scientists undertook a manic search for this gene, screening every species they could get their hands on. I, for one, Bishop acknowledged, failed to foresee the eventual outcome. They explored the DNA of ducks, turkeys, geese, even one of the world's largest and most primitive birds, the flightless Australian emu, and encountered startling results. All had the gene. They looked at the cells of mice, cows, rabbits, and fish. The gene that was once thought of as a chicken gene was in fact present in every one of these species. Finally, they screened human DNA and were excited to find it there as well. The researchers were startled by the vigorous skepticism of some whose incredulous response was, "Are you trying to tell us that a chicken gene is also in humans?" Bishop was flabbergasted by this biological naivete on the part of accomplished scientists. Had Darwin, he wondered, labored in vain? By now, reality was staring them in the face. Clearly, the gene is present throughout the evolutionary scale, having persisted for 500 million years, and belongs to the normal genome. The gene is not inherently cancer-causing, but functions as a regular part of the cellular machinery, probably in connection with regulation of its development or growth. Retroviruses picked up these normal cellular genes and instigated changes that caused them to become cancerous. Such viruses thus carry a mutated cancer-promoting gene donated by a previously infected cell and may trigger the process of cancer when they infect other cells. The Rouse virus, through an accident of nature during the course of viral propagation, served as the vector for the cancer-causing genetic hitchhiker, originating in a cell. From these findings, wrote Varmus, we drew conclusions that seem even bolder in retrospect, knowing they are correct than they did at the time. Their new model points to the cellular origin of cancer-causing genes. When the ordinary gene malfunctions, it is transformed into an oncogene that can cause tumor formation. Onco stems from the Greek onkos, meaning mass or bulk. A whole new paradigm was identified. Oncogenes are normal cellular genes that control cellular growth and when mutated become cancer-causing. Thus, in each cell there is a potential time bomb, latent cancer genes that may be activated immediately or many years later. Mutations may set off the time bomb, directing one renegade cell to over-multiply and become malignant. Cancer represents a series of events at the level of the genome. Susceptibility is sometimes genetic and heritable, but cancer can also arise through chance or more often when promoted by chemical and mutagenic carcinogens or retroviruses. Bishop and Varmus shared the Nobel Prize in 1989, only 13 years after their major discovery. Their new model of the oncogene is a milestone that probes the ultimate origins of cancer, directing attention to a site deep within the cell. Their totally unexpected finding triggered a revolution in cancer research that continues today. It set in motion an avalanche of research on fundamental factors that govern the normal growth of cells and new insights into the complex group of diseases that we call cancer. It led to the discovery of scores of different oncogenes and their functioning through protein messengers that offer targets for drug treatment and someday, it is anticipated, ways of preventing cancer altogether. like riding a bicycle. In 1993, Varmus described how strongly research gripped him. It's an addiction, it's a drug, it's a craving, I have to have it. A long-distance bicyclist, Varmus drew a compelling metaphor for scientific research from this activity. Long, flat intervals, steep, sweaty, even competitive climbs, an occasional cresting of a mountain pass with the triumphal downhill coast. Always work, sometimes pain, rare exhilaration, delicious fatigue and well-earned rests. After winning the Nobel Prize, Varmus became director of the National Institutes of Health for a number of years, and then went on to head the Memorial Sloan Kettering Cancer Center in New York.